My name is Martin Messerle. I'm a professor and group leader in the Institute of Virology at Hannover Medical School. My group is interested in the interaction of cytomegalovirus with their host cells. Cytomegaloviruses are herpes viruses, and they can establish persistent infections. So that means one cannot get rid of this virus or the virus genomes after the acute infection. The virus genomes persist in the host and occasionally can come to reactivation. In particular risk groups, for example, transplant patients or unborn children, cytomegalovirus can lead to severe complications, sometimes life-threatening. There are only few antiviral drugs available to combat the infection, and also the development of a vaccine is lagging behind. We want to understand the interaction of viral proteins and cellular proteins in order to provide the basis for the development of new therapeutic options. In the excellence cluster, we investigate the so-called encapsulation network. So this is a protein complex or a number of proteins that are involved in the packaging of the viral genome into preformed capsids. We are interested how the DNA genome is spooled into the capsids and how finally the filled DNA capsids are then sealed. In previous work, we have identified a number of proteins that are involved in this process. Now we want to understand how exactly these proteins mediate this process. In other words, we want to understand how these different proteins interact with each other. By defining the interface of these proteins, we also want to identify targets for antiviral drugs, because then if we know how we can disrupt these interactions, we can then probably interrupt the replication of the virus. So what is fascinating for me is that we can collaborate with colleagues that have different expertise. For our project, it's especially important to have the colleagues that are experts in microscopy techniques and also having the possibility to acquire structural information of the proteins. Another important aspect is that we can study different herpes viruses and compare the function of related proteins. And so from the evolutionary point of view, we can learn which proteins retain their function and which changed it. And I think that may help a lot to define bottlenecks of the life cycle of the viruses, and then these bottlenecks are definitely then the targets for antiviral drugs. Nowadays we have tools and techniques to visualize proteins. And for example, cryo-electron microscopy has now a resolution to identify almost the individual amino acid. And that's really fascinating how well we can see the proteins or the protein domains. I always say seeing is believing. At the same time, we have developed techniques to mutate amino acids. One has to say in the context of the virus, not just the individual protein, but really in the context of the whole complement of the viral proteins. And so we can study by mutating individual amino acids or individual proteins, we can investigate how protein domains participate in this process of genome packaging.